Yeah, so it's only over the past year that I've been involved with the All of Us research program. And my goal today is really to tell you about the current status of this Herculean effort by the National Institutes of Health, and to talk a little bit about what data is currently available and how you can access this data. What I'd like to do first is play a video, which I think is really very nicely done. And Adria, I think I need you to...
We are one nation, one people. When called upon to give from within, we come together and find that our capacity to help others is limitless. Here, we are fearless. What lies inside all of us is more than data. It's life. It's more than insight and medical research. It's vision and honor and compassion. What's flowing through America's veins is its diversity. The next great breakthrough will be found in each and every one of us. And what we find there will unlock mysteries and eradicate disease. We ask for 1 million individuals to come forward and stand on this landmark in history. We ask America to do, once again, what she has always done. Lead the way forward. We're one nation, one people, but all of us are different, and it's those very differences that will lead to answers for generations to come.
Yeah, I think oh, there we go. Perfect. Okay. So I thought that was very nicely done. So this is a Herculean project initiated by the NIH to invite up to 1 million Americans from all different communities, and that's a really very important part of this program, to contribute data to a data set that will be available to all researchers throughout this country for discovery about what makes us healthy and what makes us sick. The timeline here gives you some idea of the planning that has gone into this project. Is there an arrow or no? There we go. So the program was first announced in 2015 during President Barack Obama's State of the Union address, where at the time they talked about the Precision Medicine Initiative cohort, and that was eventually renamed the All of Us program. You can see July 6, 2016, some of the first centers that were piloting this program received funding. There was extensive beta testing that had to be done in several states. And then finally, in May of 2018, there was a national launch of the program. At that point, there was about 30,000 people enrolled in the All of Us program. In 2019, there was a data browser that was launched, and I'm going to go through the data browser with you so that if you want to see what kind of data is available in All of Us for potential projects, that's sort of part of my role today. All right. So I will tell you that the program is 50% there. They reached 500,000 participants several months ago, which is really, really quite remarkable. As you might imagine, the pandemic has affected every single aspect of our lives, including the enrollment in the All of Us program. So you can look at this graph and how nicely the enrollment was going, and then boom, it's almost plateaued. And we're coming out of it, but still the slope of this is not quite where it was beforehand. So it's taking time to get back where we were. This really summarizes the goals of this program. It's trying to engage, community engagement, like Dr. Lori Carter Edwards said yesterday, has been an enormous aspect of this program. And the NIH has spent huge amounts of money on community engagement. And you will see when I talk to you about the distribution of the data, they've done a pretty good job at engaging underrepresented groups in biomedical research. We're also going to take a look at the data. There's biologic data, there's social data, there's genomic data. And so it's a rich source for discovery. And they want to make this data easily accessible to anyone who wants to use it. Moreover, these participants are going to be followed longitudinally for at least 10 years. It's a bit of a challenge to retain people in the program once they've given all of their information, to continue to get information, but that's, again, one of the challenges of the program. So how do people enroll in the All of Us program? I don't know, some of you might be enrolled in the program, but there's two main ways. One is there are health care provider organizations throughout the country, and these are usually academic medical centers, and you will see I'll talk a little bit about the SouthEast Enrollment Center as well as the South Enrollment Center. These places, everyone starts online to register and then makes an appointment at one of these healthcare provider organizations. And during that appointment, there's lots of data that's acquired, and I'll go over that in a moment. And then there's direct volunteers at other places. So, for example, Emory is part of the SouthEast Enrollment Center, and this enrollment center was spearheaded by the folks at Miami, in the University of Miami. Stefan Zuckner is the PI of the SouthEast Enrollment Center, but it also includes the University of Florida, and then they reached out to us in Georgia. So it's Emory, under the PI is Tracy Rosser, and then we have Morehouse that has joined us, and recently Puerto Rico, as you know, which is part of the SERGG conglomerate, has joined the SouthEast Enrollment Center. So we all provide places for enrollment for participants in the All of Us program. Now, if we want to look at the SERGG area, there is a southern network which includes Tulane, UAB, and many other academic centers in the southern network. But also there are some places where people can enroll in North Carolina, Tennessee, South Carolina, and Mississippi. Some of these places are not as comprehensive as far as the data that's obtained, but people can still submit samples. And importantly, Vanderbilt serves as the main data and informatics repository for the entire program. They maintain the web-based research portal for this program. So I wanted to show you this slide because it gives you some idea of enrollment by geography. You'll notice that the darker blue is higher numbers from that particular region. And you'll notice that Arizona and California are quite blue. In Arizona, you might sort of wonder, like, what's that about? So it turns out that the program was piloted in Arizona was one of the states where it was piloted in. And they've done an excellent job at reaching out to underrepresented communities. And you see the Southeast region is actually doing quite well. And then, of course, just based on numbers, you'll see the Northeast is quite high, as well as areas in the Midwest. Now, this bar graph shows you the current distribution of participants by race and ethnicity. And what I also did was take the 2020 census data and put it underneath this bar graph. And so it's actually pretty impressive. So if you look here, 48.9% white in the All of Us data set. And if you come down here to white alone, not Hispanic or Latino, it's 59%. So pretty good. Here, Asian, you go down to the census, Asian is 6%. So here we're 3%. And the program understands this, they follow this, and there is actually a concentrated effort to try to increase Asian participation in the program. Importantly, if you come to here, Black, African American, or African, 19.9%. And if you go to Black or African American, 13.6%. That's pretty good. I was quite impressed when I looked at this data. And then, if you go to Hispanic, Latino, or Spanish, you have 16.8%. And if this is taxing my... now here we are. It's 18.9%. So, again, pretty good. So I think the program is really making an effort to invite and partner with underserved communities. More importantly, well, not more importantly, just as important, communities of different sexual orientations. So the LGBTQ plus communities have been reached out to, and these are another underrepresented in biomedical research. And currently, the All of Us program has about 30,000 participants who identify in the LGBTQ plus communities. So again, they really are making an effort to embrace as many communities as possible. If we look at the age of the participants, you will notice that there is a more mature, rather than older, more mature number of participants in the program, and that's sort of understandable. Currently, only 18 and older are allowed to enroll in All of Us. There is a plan to open the program to pediatric enrollment in 2023, and they want to target birth to 5 years of age. This is going to be a challenge. There's another reason I think they asked me to be on the program, since I'm a pediatrician by training and also work a lot with the Children's Hospital. So we've already partnered with our children's hospitals to try to figure out the best way to try to enroll children into the All of Us program. Okay, so, oops, sorry. So what do the participants get out of this program? So an opportunity to improve health of future generations, a chance to learn about their own health, they can access their own data, ensure that their particular community is included in biological studies, learn about additional research opportunities, choice to meet others, and they try to stress that this is a long-term relationship. There's actually been an ancillary study that has come out of the All of Us program. This will be launched next year. It's a nutrition program for precision health, and it's pretty ambitious, actually. 12,000 participants who are already within the program, they're going to have their baseline diet and physiologic response to meal challenges done. And then of this group, 1,500 to 2,000, they'll do an intervention, and then 500 to 1,000 of those, they'll do another type of intervention. And they're going to collect tons of data, including microbiome data, physiologic, metabolic, behavioral, cognitive, etc. So I think once this data comes out, it could be quite revealing about how diet influences health amongst different populations. Not really sure what time it is, but at any rate. So I wanted to talk a little bit about the data that's available and how you can at least preliminarily look at some of the data that's available. So all participants are consented, and a lot of this is done online, and they give authorization to access their electronic health records. Now, of course, all this information is de-identified, okay? And then there are a number of surveys that the participants are asked to answer. And these include things about lifestyle, overall health, family medical history, and there's some new social aspects of health that have been added to the surveys. If participants go to a particular center for an appointment, they will have physical measurements done, which include blood pressure, heart rate, height, weight, BMI, hip circumference, and waist circumference. Then they will provide samples. Typically, it's blood. In the time of COVID, saliva has been used. Blood is preferred because they also isolate plasma for other types of studies, which you'll see in a moment. Urine, you don't have to give a liter, like Dr. Edwards was saying yesterday, but they do collect urine. And these are all sent to Mayo. Mayo is the center that processes all of this, and I believe stores it all there. And then DNA, of course, is isolated and sent to the sequencing centers. And then there's a small group that are wearing Fitbits for continuous data monitoring. The genomics plans, there's 3 main genomics centers, and then there's several other centers that help with quality control. The Broad, I think, and University of Washington are 2 of the main centers. I'm not sure which the third one, but at any rate, the plans are to do genome sequencing, also some genotype, some array genotyping of all patients, not patients, participants. Return of information, this has kind of been an area where I don't think the program has been very good about yet. Color, as you know, as you may know, is the genetic counseling company that has been embraced by the program to provide counseling for folks who get return of information with medically actionable results. In general, the return of genomic results has been slow, and I think what a lot of people have found is they get information like, you know, you're half Swedish and half Spanish, and things like cilantro is going to taste like soap to you. And they've not been very excited about that data. And I think part of the problem that the program has is the pre-test counseling about genomic sequencing has not been where it should be. So personally, as a geneticist, I think that's a problem, but the program will need to deal with that. It's important to realize that for each participant, all the different data types are not necessarily available, right? So ideally, there will be sequencing for all patients, but there's only a subset of patients who will have sequencing and Fitbit data. Right? There's only a percentage of patients that will have electronic health record information, the measurements, as well as the surveys filled out. And there's sort of no way a priori to know unless you have a project that you want to do and you're going to go deep into the data. So again, the one of the main goals of the program are to make all of this data accessible to anyone in here or anyone in any other place who wants to use this data to answer a question. And currently, there's over 2,000 registered researchers. There's over 2,000 projects, and I'll show you how to access those. And that represents over 300 institutions. What kind of questions you could ask? Infectious diseases is one example. Does infection with a neurotropic virus increase the risk of neurodegenerative disease later in life? What are the social factors associated with vaccination rates, right? Something that is on everybody's mind, really, these days. And so, depending upon what you're interested in, what groups you want to look at, there may be data there that could help answer questions and discover new concepts about health and disease. So again, the program really wants to create a demographically diverse researcher cohort, encourage students to access the database and early stage investigators and the like. So with that, how do you look at the data? So you can go to the Research Hub and do this. Okay, the data access is tiered. So there's a public access that we're going to go through some aspects of that right now that anyone could look at and get some idea if there's information there that you might be interested in. If you have a specific project that you want to do, a specific question or 2 or 3 questions that are related, you need to apply for the registered tier. And if you want to use the genomic data, you need to apply for a control tier. And it's not hard to do that, right? Your institution needs to be registered with all of us. And I will tell you, I looked. Almost every institution represented at this meeting is registered by all of us. There's just a couple that are not. And if you wanted to and your institution is not registered, it's quite easy to get that done. All right. So this is really the first page of the data browser. And you can look at the electronic health records, and you could do a search on conditions, like what medical conditions are in there. You can also look at drug interactions. You could look at laboratory values. You can look at some of the surveys down here. And so let's take a look at conditions. Okay, so if you just click on this, what will come up is the most common conditions, right? And so this is really not surprising. You'll see essential hypertension. You'll see hyperlipidemia. You'll see chest pain, cough, abdominal pain, chronic pain, all common conditions. For people in this room, we might be interested in something that's a little bit more rare than some of these, right? Since most of us do rare disease work. So I did a search on neurofibromatosis. Just what the heck. And it turns out there's 160 participants in the database with NF1. And that's about a prevalence of 0.07%, and the Finns estimate NF1 at about 0.5%. So if you're interested in NF1, whatever aspects of NF1, you may be able to actually get something, publishable out of this. Make some interesting findings. Since we're going to have a talk later on amyloidosis, I thought, let me look at amyloidosis. And there's greater than 400 participants in the program with amyloidosis. So if you were to think of a project that you wanted to do with amyloidosis and you had specific questions you wanted to ask, right, you would then register, right? You would submit your research questions, the data that you want to access, and get approval. And then you would go ahead and go from there. What about laboratory measurements? So I clicked on that just to see. And, as you might imagine, you see the components of a Chem7, sodium, potassium. Okay. Not that interesting, right? Most of us do genetics. So I thought, well, what might I want to look at? Homocysteine, right? So I put homocysteine in, and, there's like 6,500 entries with homocysteine. So, you could think of a project like what percentage of these people had a stroke? You could then try to access the genomic data. Are there variants in the genes involved in homocysteine metabolism, right? There's a whole bunch of things you could think about doing. So those are those. There's currently over 100,000 genome sequences. If you click on this, you then put in your gene of interest. So, of course, because it's genomic sequencing, there's a hell of a lot of variants in there. So you probably need some help analyzing these. But at any rate, I put in NF1, and as you can see, there's 56,000 variants in there. You can pick the variant that you're interested in, and they'll give you the allele frequency in the all of us, the various all of us groups. So there's a lot of genomic data that's available for people to analyze. So if you do go to that tier 2 or 3 level of analysis, there are a number of tools that are available. And honestly, I don't know all the details of these tools because I don't have a project in here yet, although quite frankly, I'm thinking of a whole bunch to do. But the one thing you will be able to do if you register a particular research project is a workspace. And that's where you can access, store, and analyze your data for your specific project. It's important to note that all of the work you do with the All of Us database remains in the cloud. You can't download the data to your local computer. It stays within All of Us. There's also notebooks where you can do analyses with programs like Python and R for highly powered statistical analysis. You can build various data sets or cohorts, like you would build a cohort of patients with with elevated homocysteine, and you might want the ones with elevated homocysteine and documented stroke, right? Something like that. And there's also a Workbench user support hub for any questions you have about using the database. So this really just summarizes it all up here. So, the participants, they enroll, their data is curated. You can take a look at the data, public access. You've got an interesting project, you register, and then you go to town. If you're interested, you can subscribe to a newsletter that All of Us has quarterly. When there's new data releases, you can be alerted to what's available. So for the last bit, I just wanted to chat about some of the projects that are currently being, that have been done using these data sets. You can go to the research projects directory, and you will see that there are over 2,000 active projects. So, for example, here you can see chronic pancreatitis, health disparities, chronic disease outcomes of patients with limited English proficiency, etc. And you could just go through this whole list. So one of the first studies that came out of the All of Us program was really quite interesting. The bottom line was, COVID was here long before, well, maybe not long before, but before people actually documented infections. And so in this particular study, so for example, in Illinois, these were patients enrolled in All of Us whose plasma was analyzed for COVID antibodies, and they were positive about a month before the first case was documented in the state. Okay, the same thing was found in Massachusetts way before the first infection was reported in the state. Same thing in Pennsylvania. Here's an All of Us patient that had SARS-CoV-2 antibodies, and this was the first patients reported. And of course, March is when all hell broke loose in everybody's lives, right? So I guess in some respects it's good, because then our lives could have been destroyed in January rather than March. So really very interesting what was done with this data set. The next project is really quite interesting, and this was done by Alvin Carrasquillo, one of the co-PIs at the University of Miami, and his colleagues. So there's something called the Latino cardiovascular disease paradox. And that means that, though many Latinos have higher rates and or poorer control of cardiovascular risk factors, when studies have looked at the prevalence of cardiovascular disease in Latinos, it's always less than non-Hispanic whites. And it just doesn't make sense, okay? Which is why they call this the Latino cardiovascular disease paradox. So what Alvin and his colleagues did was they used the All of Us data set, because it's actually the largest Latino and Hispanic data set available. And when they did that, what they found was, when you look at women, Latino women had twice the rate of cardiovascular disease than white women, and a little bit less than black non-Latino women. And when you looked at men, they actually had a rate higher than black non-Latino and white non-Latino. So based on the All of Us data set, the Latino paradox is probably not true. Now, clearly it needs to be validated with other data sets, but it's really quite striking what can be discovered with these data sets. And then the last example I wanted to go over is a recent one, again, that validates the database to study a disorder such as atrial fibrillation. And this was done by Alvaro Alonso at the Rollins School of Public Health, where they just looked at atrial fibrillation in about 92,000 participants. And they found that 4.2% had atrial fibrillation at the time of enrollment. This is a little higher than the general population prevalence of 1 to 2%, but that's because of the more mature participant distribution in the All of Us data set. What they also found was that there was a higher prevalence with older age, which is well known. As you can see, as you go up in age, the prevalence goes up 80 to 89. It's 19%, right? Greater in men than women, that's already been shown before. Highest in whites, that's also been shown before. And so he validated the All of Us data set to continue to study atrial fibrillation. And so they're looking at other factors that contribute to this problem. To end with, there are two clips where these researchers talk about the utility of the data sets. And so I just thought we would play each of these, and then we could open it up for questions. Adria, do you want? Yeah, perfect.
I am really interested in using data, and specifically electronic health record data, for understanding risk stratification and precision medicine in ophthalmology. I got interested in using the All of Us database because of the wealth of electronic health record data. And another aspect I was very interested in was the diversity of the patients and individuals who are represented in the database. With All of Us, because there are data from so many different enrollment sites, but it's all gone through this very rigorous de-identification protocol. I think it makes it so much easier for researchers to be able to have access to data that can serve as their external validation cohort. One of the great things I think that All of Us has provided is a really great overview of what the workbench looks like, as well as a publicly available data browser that's available to anyone with an internet connection, essentially. A lot of eye conditions disproportionately affect women and minorities. And so understanding how these diseases progress and what the risk factors are and what treatments are most effective in these different groups is really important. In many cases, it's the social determinants rather than specific clinical, comorbidities or medications even that can influence that person's ultimate outcome. So this was a model predicting the risk of progressing towards glaucoma surgical intervention. And the initial model, I had used data from a single institution, my home institution. And so we had a smaller sample size than what was available in All of Us. We had generated some models and it seemed to show some signal where the systemic medications and comorbidities had some predictive value in identifying high-risk patients. And so my goal with All of Us was to say, okay, well, what if we used this larger, more diverse samples available in All of Us to really validate this model and see if we can either increase predictive performance or show that these predictors are important, and really to see whether we have a generalizable result. We hope to build upon that work and further refine our understanding of systemic risk factors with glaucoma progression, and hopefully build a predictive model that'll help clinicians better identify those patients who may be higher risk because of those systemic risk factors. I always encourage people to look at the data browser and the Survey Explorer, and then if they are interested, to go through the steps in joining as an All of Us user. And I usually direct people to the tutorials and the videos, as well as the frequently asked questions section to help to get them oriented to the interface. Through the demonstration project experience, I did meet a lot of other researchers, and it was great to, learn from them as far as how do you actually do the phenotyping? How are you defining your cohorts? How do you work with, the OMOP common data model that underlies All of Us? Even after we published the paper describing our first analysis in All of Us, I actually did have someone reach out to me from a different institution and say, do you mind if we take a look at your code? We'd like to do some similar work here. And was really easy in the system because they were already a registered All of Us user. I could just add them to our notebooks very easily. I wouldn't have had that opportunity necessarily before had it not been for All of Us.
Maybe we'll just stop with that one. He talks about pharmacogenomics. Same thing. Same story. So, so that's really, Adria, I think I need you to advance it because, shut her up. So yeah. So basically, hopefully I've given you some idea of the richness of this database, how you can access it, what are some of the tools that are available, and you certainly can reach out to us at Emory, and it's important also to realize that there are a huge number of partners that are involved in the All of Us program, in particular that reinforces the community engagement aspects of this really Herculean project. So And I'm happy to answer any questions.